Hello, welcome to the next video. This is all going to cover blood typing, so it's a little bit of a continuation of red blood cells, but it's more applicable, kind of a clinical aspect of red blood cells. So just kind of a forewarning is blood typing can be kind of confusing. It may seem pretty straightforward at the beginning, but then when you try to apply it and kind of do transfusion problems or figure out who can donate to what and all that kind of stuff, people can get stuck or confused, mixed fused is what I like to say, fairly easily. So one of the benefits of doing these lecture recordings is that you as a student can go back and watch it and pause it and rewind it and watch it as many times as you need to, where if we were to do an in-person class, we get the one day to cover it. So take advantage of this ability to rewatch this discussion as many times as you need to try to get your understanding of blood typing down pat, so you can continue to answer questions clearly and understand what's going on. Because if you don't, if you're just kind of staying at that superficial understanding, it's going to be hard and it'll be easily mixed fused if you don't try to thoroughly understand blood typing. But I'm going to try to take it step by step to walk you through all of this information. All right, so I usually like to present this as like a foundation. We have different levels and we're going to start very basic and then we're going to move up and add a layer of complexity and so forth. So by the time we get to all of the information that we need, we will have kind of built everything slowly in our foundation. So before we even get to blood typing, I need to introduce a couple words. We've got quite a few A words associated with blood typing. So our first one is an antigen. What is an antigen? An antigen is a surface protein. All cells have them, not just red blood cells, white blood cells, bacteria, fat cells, muscle cells, surface proteins. Think of a phospholipid bilayer when you're learning about membrane anatomy back in general bio. We had peripheral proteins and integral proteins and ion channels and glycoproteins. We had all of these things stuck into that phospholipid bilayer. Those are antigens. That's all it is. We're just giving a special name. And the reason why they're called antigens is because they can generate antibodies. That's where antigen comes from. So antigens are surface proteins, okay? The other A word that we're going to be seeing is called an antibody. So antibodies are globulins. So kind of going back to our composition of blood lecture, antibodies fall under the globulin protein category of plasma proteins. So these are plasma proteins. They are found floating in the blood. Okay, so we're going to see where we find the antigens and what types we have, and where we're going to find the antibodies and what types we have, and how antigens and antibodies can react to cause some problems inside of the body. So first, we are going to take a look at antigens. That's our first level. Okay, this is our foundation of blood typing, is identifying the antigens. Now, again, like I said, I'm only going to do the A and B. I'm not going to do RH because I want to get the A and B down before we add something different and new. So in this picture, on the surface of the red blood cells, the little red circles are our A antigens. So that they're kind of showing it as a geometric shape. So if your genes code for the A antigen on the surface of your cell, you would be type A blood. The blue triangles are the B antigen. Okay. And another kind of reminiscing back to Bite 211 is our genetics. And if you remember the idea of codominance and multiple alleles, ABO blood types falls under both of those categories. So A and B are codominant to each other. So if you have both A and B antigens, you are considered type AB blood because one does not override the other. So you have both antigens. The other option is you have neither. So you don't have any antigens. Your surface is, for these particular antigens, your surface is blank. You're going to have other antigens, but we're not going to talk about those. So for antigens, these are your options. If you have A antigens, you're type A blood. If you have B antigens, you're type B blood. If you have both antigens, you're type AB blood. And if you don't have either, you are type O. Okay, so that's our foundation number one, layer number one, pretty straightforward, okay? A, B, and O. Okay. Now let's take a look at the plasma proteins. These are the antibodies. Now, if I were to draw an antibody, it's kind of this Y-shaped molecule. I know they show the like 5 Ys, and that's fine. But if I had this particular antibody coming into contact with this particular antigen, do those molecular shapes fit? No. So the A antigen is a circle. I have the binding site on this antibody is a triangle. So this would actually be, if I were to label it, this would be an anti-B antibody because it would recognize the B antigen. It ignores the A antigen. So when we're trying to partner up our blood cell surface antigens with our blood plasma antibodies, we do not want the same combination. We don't want anti-A antibodies if we have A antigens, because then you're going to get what's called agglutination, which is all this clumping and destruction of the cells, which can kill you, right? So we don't want that to happen. So if you have the A antigen, you will have anti-B antibodies. If you have the B antigen, you will have anti-A antibodies. If you have both antibodies or antigens, you will have neither, if I can spell it correctly. If you have neither antigens, oh my gosh, why do I keep wanting to put a G in neither? You're going to have both antibodies, right? So we can see that in these combination pictures here. So type A antigens, anti-B antibodies. Type B antigens, anti-A antibodies. Both AB antigens, neither AB antibodies. Neither AB antigens, both anti-A and anti-B antibodies. Okay, so that's layer 2. So we've got antigens on the surface give us our blood type. Antibodies in the plasma recognize the opposite type, right? So if you have type A blood, you'd be able to recognize B antigens. If you have type B blood, you're able to recognize A antigens. If you have AB blood, you can't recognize any blood type. If you are type O, you can recognize both A and B. Okay, so let that sink in. I'm going to go through a couple of examples on the next slide. So what's the point? What's the big deal for this? Well, the big deal is if you do mix the same antigen with its matching antibody, what you get is what's called agglutination, which is another A word down here, which is basically clumping. So the antibody that matches its antigen binds to it, and because these antibodies are kind of multi-edged and starburst, you can get a whole bunch of red blood cells clumped and stuck together, and then ultimately it causes the blood cells to lyse and be destroyed. It destroys your red blood cells, which is bad, plus you get all this cell debris and clumped blood trying to fit through your capillaries, which is also bad, and you end up dying or having a very severe reaction that needs immediate medical attention. So that's why we have the opposites kind of found in your blood. So let's take a look at do some practice problems, and I do encourage you to try to come up with your own practice problems to work through to give you that confidence that you can answer questions about blood typing. But I'm going to give you a rule that should help make it easier. So I want you to always look at the donor's antigens and the recipient's antibodies. Okay, so keep that in mind. Donor's antigens, because if you are a donor, you're giving your whole pint or unit of blood with all of your red blood cells filled and covered with all of your antigens into the recipient who has 5 to 6 liters of blood plasma, majority of it blood plasma, with all of those antibodies that can have a direct impact and recognize those antigens. So that's the major problem, is when the donor's antigens are recognized by the recipient's antibodies. So let's throw some blood types up there. I usually like to set it up in a column of donors on one side and recipients on the other. Okay, and let's just do, let's say if the donor is O and the recipient is B. Okay, so at the beginning, when you're first learning all of these things, I encourage you to write down all the information you know about each blood type and then apply our rule here. So this has neither antigen, but it has both antibodies. Type B has B antigens and anti-A antibodies. Okay, so now let's go and take a look at our rule. Look at the donor's antigens. Okay, so I'll circle that, and the recipient's antibodies. Okay, so I'll circle that. So if you're a donor, you of O type, you have no antigens on your surface. If the recipient is B, it's going to have anti-A antibodies. So anti-A antibodies can't recognize any O blood type, so that's okay. That's totally fine. What if we did type A blood into AB blood? Okay, can we do that? Well, let's write it down. So we have A antigens, anti-B antibodies. We have A and B antigens and neither antibodies. Okay. All right, so let's follow our rule. Look at the donor's antigens and the recipient's antibodies. All right, so here's another case where we have the recipient doesn't have any antibodies to recognize any of the A antigens coming in. That's fine. Okay, let's do one more and then we'll add another layer of complexity. What if we were to do B blood? No, we'll do A blood again into O. I gotta move it over because my head's taking up too much space. All right, so let's write down what we know. A antigens, anti-B antibodies, neither antigen, both antibodies. Okay, so let's look at our rules. Look at the donor's antigens. Okay, donor's antigens, recipient's antibodies. They have both anti-A and anti-B. So if you are a blood type O, you can recognize both A and B blood, and somebody dumps in a whole unit of blood that has full of A antigens, there's going to be a problem. So A blood cannot give to O blood, but O blood can give to pretty much anybody. So sometimes O has been considered a universal donor because it doesn't have any antigens to recognize, and AB blood is sometimes called the universal recipient because it doesn't have any antibodies to recognize anybody's antigens. So that might be another way to remember our little saying up here. So keep that in mind. Look at the donor's antigens and the recipient's antibodies. All right, so again, I encourage you to set up some of these types of problems yourself to make sure you're comfortable with these relationships. Now we're going to add the RH factor, the positive and negative blood types. Okay, so we're going to use this example of a pregnancy, a couple pregnancies, but first, before we do that, I got to talk about what the RH factor is. The RH factor is just another antibody. So now antigen. So now we had A, B, and now our third option is RH. So it's genetically predetermined. RH is dominant to no RH. So A and B are co-dominant to each other, but dominant over the O genotype. RH is dominant to no RH. So you could have A positive, A negative, B positive, B negative, AB positive, AB negative, and O positive and O negative. So you have a positive negative option for each of the 4 blood types we just took a look at. So let's kind of visually see what the difference is between a positive and a negative blood type. So let's take A, we'll do 2 red blood cells, okay? And I'll just write the letter A for the antigen. Okay, so type A, you're always going to have A antigens. That's not going to change. Okay. If you are A positive, you will also have positive antigens on your surface. So this would be an A positive blood type. If you are A negative, you don't. You just have your A antigens on the surface. Well, what about our antibodies? So if you're A blood type, you're going to have anti-B. That's going to be true for both. But now, do we have antibodies against the RH factor? That's where it gets a little bit tricky. If you are A positive, you will never have RH antibodies because you never want to have an antibody that would recognize your surface antigens. So A positive will never have Rh antibodies, anti-Rh antibodies. But A negative might, because if you're A negative, you don't have Rh and you maybe want the ability to recognize an Rh positive blood. But you are not born with anti-Rh antibodies. They only show up after what we call sensitization. So if you are a negative blood type, and this is only an issue for negative blood types, only if you are exposed to a positive blood type, your body, it's almost like an immune response, will then produce anti-Rh antigens, antibodies, only after you've been exposed. If you have a negative blood type and you never are exposed to anybody else's blood in your entire lifetime, you will never produce anti-Rh antibodies. It won't even be an issue. It is only an issue for negative blood type individuals who've been exposed to positive blood type. Now, it's not very common for people to be exposed to somebody else's blood being a positive blood type at that. So blood transfusions, accidents, those types of things. Probably the most common way that a negative blood type can be exposed to a positive blood type is through pregnancy, because mom and baby's blood does not have to be the same, because the blood does not cross the placenta. The placenta is a good enough barrier that your anti-A and anti-B antibodies do not cross. The whole big red blood cells do not cross. So because this is a genetic thing, let's say we have a mom that has AO genotype, so she's heterozygous, and both of her alleles are negative, so she has no Rh factor, but she mates with a man that is also heterozygous. Let's make him homozygous A. And also homozygous Rh. So if they mate, these are the possible blood types their kids can have. Get rid of that. Do it properly. So what that means is there is a 100% chance that their kid, all of their kids are going to be A positive. That's the only blood type that they can produce. So A negative mom mating with an A positive dad, 100% of the kids are going to be A positive. And that's okay. I mean, that is, you're not going to have any problem unless they have more than one kid. Okay. So let's take a look at what happens. So let's say A negative mom, right? So here's our A negative mom. Gets pregnant, has the baby. The baby can only be A positive. So baby's A positive. During the entire pregnancy, there's no mixing of the blood. Okay? During delivery, though, there can be, because the placenta kind of gets pulled away from the wall of the uterus. There's mom's blood vessels are open, baby's blood vessels are open. You can get some mixing of the blood during delivery. Some bleeding, hemorrhaging kind of happens. So now the A negative blood has A positive blood kind of coming up and mingling in it. So now mom has been exposed to this positive blood type. So it does not change her blood type. She is still A negative. Okay? She still has A antigens. She's not doing anything to her blood type, but what is happening is a change in her antibodies. So this is what she was always. A antigens on the surface, no Rh, anti-B antibodies, that's totally fine. However, after that exposure, her body is saying, hey, that's something new. I need to recognize that Rh factor in the future. So then it starts producing the anti-Rh antibodies. So now mom, still A negative, still has anti-B antibodies, but is now circulating anti-Rh antibodies. Okay, if she just had one kid, that's not going to be a problem. But if she has a second kid, which according to our scenario is going to be positive because that's the only type of offspring that they can produce, these anti-Rh antibodies are smaller than the anti-A and anti-B. They can cross the placenta. And so mom's new anti-Rh antibodies crosses the placenta, attacks baby's red blood cells, and destroys the red blood cells. This is called fetal hemolytic anemia because it usually happens during the fetus and fetal development, and sometimes the baby can die before it's even born, or the baby can die early or be born prematurely and have severe anemia because most of their red blood cells have been destroyed. So like the mom's body, antibodies are attacking the baby's red blood cells. But that doesn't happen very often because it can only happen in very specific scenarios. One, the mom has to be a negative blood type. Two, she has to have been exposed at some point in her life to a positive blood type. And 3, the mate that she is mating with has to donate a positive blood type, because if the mom's negative and the dad's negative, it's not going to be a problem. If the mom's positive and the dad's positive, it's not going to be a problem. If the mom's negative and the dad's positive on his blood type, and she's been exposed or had multiple pregnancies, then that's when there's a problem. So it kind of focuses it and narrows it down on those particular conditions. All right, so this is our top level of complexity of our blood typing. We have antigens, antibodies, who goes where. Now I have adding an Rh antigen and a selective antibody production based on sensitivity and exposure. So the last slide here, I just want to do some more practice questions, now adding the positive and negative to it. So again, like I did before, let's remind ourselves to look at, look at donors, antigens, and recipients, antibodies. Okay, so let's set up our 2 columns here, donor and recipient. All right, so let's do type O positive to B negative. Okay, so let's see what we get. Let's write down what we know about our blood types. So we have neither A or B antigen, but we have a Rh antigen, and we have both anti-A and anti-B antibodies. For type B, we have B antigens and anti-A antibodies. Now, what about anti-Rh? I would have to tell you about the person. I would have to say sensitized or non-sensitized. So let's have this be a sensitized person. Let's say this person sensitized. Let's say this person has been sensitized. So what that means is now they also have anti-Rh antibodies as well as their anti-A. So like our kind of rule says, donors antigens, recipients antibodies. So donors antigens, really they only have the Rh antigen because the O doesn't provide any antigens, and the recipient's antibodies. Okay? Well, the anti-A is not going to cause a problem because there's neither A or B antigens on the O, but the anti-Rh will cause a problem. So this is a bad scenario because the recipient was a sensitized negative, they cannot receive positive blood anymore. Let's say it wasn't. Let's say it wasn't a sensitized B negative, and this was the information. Donors antibodies. Could a B negative unsensitized person receive positive blood type? Yes, once. Because once they receive it, then they would become sensitized and they would no longer be able to receive that positive blood anymore. Let's do one more. Let's do AB negative giving to B positive. I'm going to have to write the stuff down below on this one. So let's have A antigens, B antigens, and neither antibodies. Maybe I can write it up here. B antigens, Rh antigens, and anti-A antibodies. No anti-Rh because it's a positive blood type. Positive blood type's never going to have anti-Rh. So donors antigens, recipients antibodies. Could recipient with anti-A antibodies recognize an AB blood type? Yes, that's bad. So that would be a bad match. All right, so that's our blood typing. Again, it's lots of layers, antigens, antibodies, Rh factors, sensitized antibody production. Please, I encourage you to keep working. You can do all the combinations if you want to. If you want to make a table to say, here's the blood type, here's who it can donate to, here's who it can receive from. That would be a great way to get all the information on one page. And then you can use that as a key to answer your questions of all of this kind of donor recipient antigen antibody stuff. All right, so that wraps up our blood typing lecture. I will see you next time for white blood cells. Bye.